Hello everyone and welcome to another demonstration video. And in today's demonstration video, we are going to look at what is docking, okay, and what are the tools using which we can pursue docking. And particularly we'll be talking about two tools. One is bioinformatics based tool called as your SwissDock. Another one is your AI/ML based tool, basically artificial intelligence and machine learning based tool. That is your using your NVIDIA server. The name of the tool is DiffDock. Okay. So in this particular demonstration, I wanted to keep it very short and crisp, okay, so that it is easier for you to iterate all of the process, okay, and you don't have to go through multiple literature and that, okay. So you can simply look at this, okay, this particular thing, and I will also try to define you the particular principle behind it, okay, in very what do you say, one or two lines, okay. So how much ever is possible. So let's see, okay. So first things first, what is docking? Okay, so basically docking is why we do docking. Okay, so basically we do docking in order to establish a relationship between two molecules. Okay, so particularly your two molecules would be interacting. Mostly you must have seen that in an enzyme kinetic reaction or likewise. Okay, so you have a, let's suppose it is just like a lock and a key kind of a model. Okay, but in actual scenario, when the things are happening inside the body, it is not always the same, okay. So you have a protein, okay, so it has multiple contours. Why do I say contours? Because it can have your pockets as well as cavities, okay. So if I have a drug molecule, okay, so that wants to bind to that particular cavity, so it can bind to anywhere, basically pocket 1 2 3 4, multiple pockets are there in the protein, okay. So it can bind itself to anywhere, and then it can show the activity, basically how firmly it is binding to that particular site does matter, okay. So that is why we are looking into that particular thing, that what kind of therapeutic intervention, basically the receptor can be a drug molecule. So that is what we are trying to simulate, basically, okay. So the simulation is, although like the whole simulation is done in molecular dynamic simulation, so that is a later on part. But in an inert environment, okay, so in an inert environment we are trying to see that how the ligand molecule, okay, is interacting with the macromolecule that is mostly protein, okay. So this particular macromolecule can be a protein, okay, or a receptor inside the body, okay, so mostly protein. And you have a ligand, initially it is called as lead, okay, but when you pass this particular lead through multiple screening process, virtual screening process, that includes your Lipinski's rule of 5, this that Pfizer rule. So drug likeness, you will be studying it for then your pharmacokinetic and pharmacodynamic studies, using your ADMET studies, that is your absorption, distribution, metabolism, excretion, and toxicity. After those, after all the screening processes, the last part of screening is your docking, because we want to see that how the two molecules are interacting between each other, okay. So the main thing is that, okay, and for that particular thing we are gonna see that what is the binding affinity score. So it is called as your binding affinity score, or also sometimes loosely it is called as your docking score, okay. So let's talk about your docking or affinity score. What is it? So basically, when you have a docking score of more than minus 5 kilocal per mole, because it is an energy function, mind you that, so that is why the unit is kilocal per mole. So basically, higher in the negative connotation, better it is, okay. So in most of the tools, so let's talk about, there are plethora of tools, although for your docking studies, okay. So you can use Pyrex, that also uses Autodock suite only, but it is automated, okay. Then you have your Autodock Vina is there. Apart from that, you have your Glide is there, that is paid from Schrodinger. Then apart from that, you can also use cloud server for your cavity search as well, like SwissDock server. So that is what I was telling you, that SwissDock server is there for your, like so that is maintained by your Swiss Institute of Bioinformatics, okay. And the next server that we are going to use would be of NVIDIA's. So in NVIDIA we have a like AI/ML based tool, so that is called as a DiffDock, okay. So in DiffDock, although the way of the calculation, the energy calculation part is a bit different, that is why there the what do you say, your interpretation of the result is also a bit different. Okay, so in all of the different tools, what happens is anything above -5 in the negative connotation is better. Let's suppose -6 kilocal per mole is a better affinity score than -5. Again, -8 is a better affinity score than your -5 and -6, and likewise -12 would be a very good affinity score. Okay, and then we can also cross validate this particular results using your postdoc analysis, using any of the visualization tools like your PyMOL, RasMol, Chimera, or your VMD, or even Discovery Studio, and likewise. Okay, so in those, what we can find that the affinity score is actually a probability based score of the energy function, that how much energy is required to displace, or basically in order to detach those particular two compounds. Okay, so obviously the firmer they are binding to each other, so more amount of energy would be required. And why it is in negative? Because it follows delta G, that is Gibbs free energy. That is why. So what do you say, your higher what do you say, higher energy state is unstable. Okay, so higher the energy state, more unstable it is. So that is why we need to have a lower energy. Okay, so the more it is in the negative connotation, better it is. Okay, so -12 is a better than your -10 kilocal per mole, likewise. Okay, for most of the cases. But when we are talking about DiffDock, so it is a bit different. Okay, because it is a AIML based tool, their ranking system and their scoring function is different. So how their scoring function is? Their scoring function is absolutely normal. How you handle mathematical things. Okay, just like that. Okay, so a higher number connotates your higher affinity. Like for instance, like if you have a -4 and -2, -2 is better in case of DiffDock. Okay, whereas in case of your Autodock Vina, what we will be demonstrating using Swiss model. Okay, sorry, SwissDock. Okay, so in that one, what happens is there it is just the different that a higher negative value is a better value. Okay, so that is about the interpretation of the results. Okay, apart from that, we can also look at how the interaction is happening, what kind of bonds the interaction is supported by. Okay, likewise it is having multiple of hydrogen bonds, hydrophobic interactions. Okay, so your covalent bonds, so those we need to look at. Okay, van der Waals forces are also there, but van der Waals forces basically are very weak forces, or basically very weak interactions, so we don't basically count upon them. Okay, so that is about it. Okay, so that is what we are gonna see in today's demonstration. Okay, so without further ado, so we'll be straight away diving into the demonstration part, because that is what you are waiting for. Okay, so I'm your host Prodyot Banerjee, and let's get started with the demonstration. Yeah, so first things first, let's have a look at SwissDock first. So in your Google or any of the search engine that you are using, okay, so you can just type in SwissDock. Okay, SwissDock. So as I have already stated that this particular site is maintained by Swiss Institute of Bioinformatics. Okay, and if you go inside this, so this is the page you will land up with. Okay, so as you can see, so you can pursue the docking via two means actually. Okay, one is your docking with attractive cavities that you see over this particular option over here. Okay, and the next option that you see is a docking with your Autodock Vina. Okay, so either of them you can choose. Okay, so both of them use a different kind of algorithm, but the most widely used in this particular world is your in the whole world mostly, although nowadays you have more of your AI based tools and automated tools. So this is also doing automatic or basically your automated docking we are trying to do. Okay, so using this particular server, so we'll be utilizing Autodock suite. Okay, so your Autodock Vina, Autodock 4, then your Pyrex, all of them follows your Autodock suite only. Okay, so Autodock is the algorithm that is doing the job in the background. Okay, so here you can see multiple of the examples that you can find over here. Okay, so you can also see those examples. Let's suppose I click on the second one. Okay, so if I click on the second one, so the protein is loaded. Apart from that, your ligand, so this is the ligand. Okay, so or basically the lead molecule, you can say the chemical molecule by large. Okay, so then this is the protein molecule. Okay, so we will look at the example. So you can have your protein, you can just clean your protein and upload. Okay, in this particular tab, or you can basically write the id pdb id of the protein over here. Okay, in this one, okay, you can write the pdb id. Okay, and after that you need to select the chain. If you want to work on all of the chain, you can click on select all. Here only one chain is there, so that is why it is showing only one a chain. Okay, apart from that, if you want to like, it will by itself, it will remove all the heteroatoms. Okay, but if you want to include any of the heteroatoms, so you can choose. So what is heteroatom in the first place? So let's talk about that. Heteroatom basically is anything which is not your protein is called as a heteroatom. Okay, so while I show you about the PDB format in a brief, so I will also tell you about what are heteroatoms basically, which are surplus basically. Okay, while doing the wet lab based experiment like X-ray crystallography, NMR, this that, there they must have included some particles of water. Apart from that, some of the metallic ions, this that, which are required for the experiment, but that is not required for us, our experiment, basically for docking purposes, because if it binds to the active site, or basically it can hinder the interaction site, or basically the active site it can interact with and hinder our process, because we are having a different ligand which we intend to bind to the cavities, okay, cavities or pockets, so that particular thing might be compromised. Okay, so that is why we need to check on this. Okay, so yeah, so if you want to include heteroatoms, you can. Okay, mostly we won't do that, okay, because heteroatoms are not of use to us. Okay, so we can continue with that, and apart from that, we have to set the arbitrary box. Okay, so based on the setting of this particular what do you say, imaginary box, so what happens is this particular box defines that the algorithm should try to fit our ligand to which particular area. Whichever area is inside this particular box will be taken into consideration. Okay, so if I like cover the whole protein, okay, so likewise I can change. Okay, so you can see when I'm changing this, okay, so it is obviously in XYZ axis. Okay, so likewise, if you can see now, it is like going in this direction. Okay, so likewise then it will go in the high direction likewise. Okay, so I can cover the whole protein. You just have to maneuver the whole thing and need to see that how much of the protein is covered or not. Okay, so if you want to do a blind docking, in that particular case you don't know the active site. Okay, so for that thing you need to cover the whole protein inside the box, so that the algorithm will try to fit the ligand in all, or like it will try to fit the ligand all across the protein, and wherever it is finding the best affinity score, or like best fit. Okay, and more number of bonds are supporting there. Okay, so based on that it will be doing the docking likewise. Okay, so that is there. So yeah, so let's see an example. Okay, so they already have 3 examples over here. We will look into one of them. Okay, so because more of customization I'm not doing, because for the beginners, so I'm not doing any customization over here. But in the second example of your AI ML based tool DiffDock, I will be like telling you from scratch that how to clean the protein, how to process the ligand. Okay, likewise we will take a arbitrary what do you say, an arbitrary example for that. Okay, so here it's needed like all of the molecules are needed to be converted to PDB-QT format. Okay, so that is the format of your docking. Okay, and how is it is to be done in actual, if you are handling your Autodock suite, Autodock Vina, or this that. Okay, so Pyrex or Autodock 4, mostly for Autodock 4 and Autodock Vina, you have to do it manually. You have to add Coulomb charges, and your or different kind of charges are there. Force field you need to do. Okay, apart from that, missing residues you need to adjust for. Apart from that, energy minimization, all those steps you would have to do manually. Okay, but here it is automated, so you just have to click on prepare ligand. Okay, and it will take some time. Okay, and then it will be prepared. See, the green tick is there, so the ligand preparation has been done, so it is converted to PDB-QT. Okay, this particular smile formula is nothing but basically it is a chemical formula for your like ligand molecule. Okay, ligand molecule is nothing but any chemical. So based on how much you are like how you are coming up with the ligand filtration process, then you can land up with a particular ligand molecule. Okay, so then, so how to know that which one is to taken for taken as a ligand? So you can go through the literature, or after doing virtual screening, because you can start with 10 lakh compounds, so that is called as your lead library preparation. After that, virtual screening happens, and which satisfies all the rules, okay, or which follows all the rules, okay, that it can be a drug or not, it passes what do you say, pharmacokinetic, pharmacodynamics, everything, okay. So in that particular case, it can be considered for, to be called as a ligand. Now you need to see that it is having a good affinity with your what do you say, the receptor you want to target it to, okay, because at the end of the day, this particular receptor would be the therapeutic intervention, okay, or drug molecule, so which we want to target against our protein, okay. So the protein, now coming on to the protein, this is the protein. So protein, you can do a protein ID. So this is a PDB ID, basically 4-letter code, it is having alphanumeric code, okay. If you go to PDB, you can easily find it, okay. Apart from that, the which particular chain you need to select, which particular chain is responsible for that particular disease or disorder, you need to know, okay. Apart from that, that particular protein should also be having a important function or role in that particular disorder, then only if we are coming up with a therapeutic intervention. Means what we want to either inhibit, like we want our ligand to act either as a, what do you say, substrate or an inhibitor, depends. Like substrate, if we are talking about antioxidant and likewise, okay, so then it can act as a substrate. Whereas if we want to confine or basically restrict the role of that particular protein, okay, so we can come up with your inhibitory molecule, so that can inhibit that particular thing, okay. Likewise, okay, so the protein preparation is also done over here, okay. So in this, all of the charges, energy minimization, all of the steps are being done over here, okay. Now, as this is a example, and I want to keep the video very short and crisp, like crisp, okay. So in that particular case, what I need to do, so I will be defining the box a very what do you say, small, okay. So although now you might be asking, so how to set the box. So the box setup, okay, so I am basically doing a site specific docking, not a blind. In the blind one, you need to cover the whole protein, mind you that, okay. So blind will take more amount of time, okay, first thing. And second thing is obviously like you only do blind docking if you don't know the active site. But for this particular protein, I already know the active site. You can go through the literature and like see the active site, and based on that you can confine the box to that particular area, that is called as your site specific. Because we know the active site, will be defining the box in that particular area, and algorithm won't try to fit itself to all other areas but only in this particular area. So that is saving out on computational power or computational rigor. It is like saving on that. So that is why it takes less time as compared to the others, okay. So I can like I will try to like do less on this, okay. So yeah, okay, you can also do this one, okay, so that the box will shift. Now you can see it has like it has come to the center, okay. So likewise, okay, so you can see so it is moving. So you can maneuver the whole protein and see how it is moving, and likewise, okay. And based on the site, you have to define, okay. Now coming on to the second fact, so that this is called as your sample exhaustiveness, okay. So what is sample exhaustiveness? As you increase this parameter, obviously your result would be more affirmed, or basically a better result you will be getting. But this includes more computational power, okay. Basically, obviously this is happening in the with the help of a supercomputer or GPU enabled computer, HPCC, high performance computing clusters, and likewise, that is working in the background. You are just leveraging that particular thing, okay, and you are being the end user of it, okay. So I can just maneuver this particular slider, okay, to lower this, or like I can go to higher numbers, okay. So more higher numbers I go to, more time it will take. It will come up with obviously better and more tangible results, okay. But now, just for the example sake, because we want to pursue the docking very fast, okay, so I will keep it in 1, okay, so that we get the results very fast, okay. Apart from that, if you are actually doing the docking, so you can increase it. All other things will remain exactly the same. It is just that it will take a bit more time, okay, because it will iterate the process that many time to be confirmed about it. It will try to cross validate its algorithm itself, okay. So it will try to fit into that particular cavity multiple times, and multiple ranking it will be doing, multiple poses it will be trying to confine it to, okay. Likewise, apart from that, anyways, for a pocket, it always gives you mostly not for a pocket, for the whole the area you have defined for that particular thing, it will do ranking. Okay, and based on that, mostly top 10 ranks it gives, okay, when you do it manually. But here mostly it gives you top 7 and top 8 likewise. Okay, so I will keep it to 1. 1 is the bare minimum, so that is what I am keeping it onto. Okay, so I will check the parameters over here. Okay, so let's see the parameters. So if it is too large, because see, as it is a cloud based server, it has some restrictions. What are those restrictions? That you cannot do too much of a large protein docking over here. Okay, so otherwise, so their server would be too much occupied, because obviously all of them are using it, all of the people in the world are using it. Okay, but most of the time it it can do it. Okay, so here you can see it is a green mark, so that obviously we can do this. And how much time it will take? Estimated 23 seconds only, because the exhaustiveness I have kept to the bare minimum possible. Okay, so likewise, okay, so the optional email address you want to give it, so you can give. So like in the background, let's suppose I increase the exhaustiveness, and if I give my mail ID, whenever the results are ready, they will mail you that the results are ready, you can download it likewise. Okay, so the enter name also you can keep it. Okay, so that is not mandatory, although optional, so it is already written over here, so we'll not go into that. Okay, so I will start with the docking. Okay, so we'll hit on start docking. Okay, so now you can see now the docking has started, so you can click on stop if you want to. Otherwise the percentage would be shown, that how much percentage it has covered or completed. Okay, so as it is going to take only few seconds, okay, 24 seconds to be precise, 23 20 second 23 24 second approximately. Okay, here you can see the job running estimate time is written over here. Okay, and this is the run time. Okay, so it is just an estimate, it has already surpassed that. As you can see, 31 second has happened. Okay, now 100% almost like almost it is done. Okay, so let's see. Okay, so yeah, so docking is finished, so they are basically redirecting us to a new page so that we can read the results of it. Okay, so here it is, here is our docking results. Okay, so based on what kind of poses it can generate, it will give me the ranks. Okay, so if I could have defined the area, like, or the particular box to a larger area, and I would have increased the exhaustiveness, so I might have gotten a good affinity score. We have got a decent one. Why? Because -5 is the bare minimum for your publication, and anything above this is considered good. Okay, so basically this is in kilocal per mole. Okay, so -5.087, okay, is the affinity score. So let's talk about this. So if I just click on this one, so because this is only the good score we are having, but mostly in like all other docking experiments, you might get -10 -9. Okay, and till -5 we will be considering. So here we have one good dock score. So let's talk about this. If I click on this, it will show me that what are the interaction like. So this is the ligand. Okay, and how you know that this is the ligand? So you can see this is the chemical molecule, and now it is interacting with this particular protein that you see. Okay, so you can point, like you can see my cursor, okay, over here. So here you can see the blue marks. Okay, so because so these are the interacting atoms. Okay, so hydrogen bonds over here are shown by the blue color. Here you can see the blue interacting atoms. So dotted what do you say lines are the bonds. Okay, so many hydrogen bonds we are finding. So okay, so the postdoc analysis also, if we can do so, although it is having a -5 affinity score, but the interaction is very good, because we are having multiple hydrogen bonds. Now let's look at the ionic interactions also, if we have any. So it would be shown in yellow, but yeah, we can't see any, so there are no ionic interactions in this. Okay, then your cation pi, if it is there, okay, so that would be shown in orange, as you can see the color. So your pi cation is also not there. Pi cation by itself is a very strong bond, covalent bond. So if it is mostly you are having a covalent bond, so the affinity score will basically go beyond -6, like -7 -8 mostly you will be having, because those are very strong bonds, so that will increase the affinity. Okay, so now coming on to hydrophobic interaction, you can see whenever I am clicking on hydrophobic interaction, here you are having a bond. Okay, So that is represented in your lighter gray, this one. Okay, so by the color connotation also you can see that which particular bond is of which type. Okay, then you have pi stacking. Okay, pi stack are there, pi alkyl stack are there, and likewise multiple types of bond are possible although. Okay, and based on whatever the bonds are, you will be having the options over here. It is not that always you will be having the same kind of option. Okay, depending upon the ligand and the protein, what kind of interaction like are being shown over there, based on that you will be having this particular representation. Okay, as you can see. Okay, and apart from that, if you want to look at where actually the protein or basically the ligand is fitting into the protein. Okay, so this is called as your cartoon representation, where you can see the alpha helices and beta sheets of the protein, but you can also go to your surface proteins like representation. Okay, this is called a surface protein representation. Here, okay, you can see that this is the protein. Okay, so this is the surface, and if I just like zoom out this one, okay, so that you can see it quite easily. Okay, so this is the whole protein. Okay, and this is fitted into this particular cavity. Okay, so it is a deep contour. Okay, or so that is why can be a cavity or a pocket likewise. Okay, so it is bounded in here. Okay, you can see this particular ligand. Okay, likewise this is the representation. Okay, so that is about it. Okay, when it comes to your like docking using your SwissDock. Okay, so now let's get on to your AI/ML based model that I was talking about, that is your DiffDock. Okay, and for DiffDock we'll be utilizing the MIT server, which is powered by NVIDIA. Okay, so you just type in like NVIDIA. Okay, NVIDIA DiffDock, DIFFDOCK. Okay, yeah. So this is the DiffDock model by MIT, Massachusetts Institute of Technology from the US. Okay, and this is maintained by your NVIDIA. Okay, so here you have it. Here also you have already an example. So you have the protein and the ligand, and here I have already told you that. So let me try to like, move this over here. Okay, so yeah, so yeah, so here the ranking would be a bit different. Here you can see the rank one is a lower number of negative, whereas there higher negative values are better affinity score. So that is the difference for of this particular, what do you say, interface. Okay, or basically this is based out of your artificial intelligence and machine learning. So this is trained like that. Okay, and it can do the job in very fast. It can do the job. Okay, and that too, like multiple, what do you say, multiple ligand docking. Apart from that, it can also do your here it can, like, you don't have to define an area, like arbitrary box is not there. Okay, in this one arbitrary box is not there and not required also, because it is so fast that it always try to do the blind docking. Okay, so blind docking means it will always include the whole protein, because what was the restriction in the whole protein, considering the whole protein, the main restriction was computational power. But here they are having like CPUs and GPUs in the background, so it is not a problem over here. Okay, so now we will look at all of the parameters, but prior to that, here we will be taking an example from scratch. Okay, how to do the docking. Okay, so because it is already having an example, so let's see what it takes. Here you can see my pointer that it supports a file format as .sdf or .mol2 in case of your ligand. Okay, and in the protein it will take .pdb. Okay, so .pdb format it will take. So then let's extract a protein from in the PDB format, and also extract a what do you say, your ligand from your any of the PubChem, Zinc, or Drug Bank, any of the tool you can use. Okay, to extract that, or any other chemical databases also you can use. Okay, so and there we will extract the ligand molecule in your .sdf or .mol format. Okay, and then we will proceed with the docking. So yeah, so let's get to PDB first. Okay, so in order to extract the protein. Okay, so yeah, okay. So internet is taking a bit time. So in the meanwhile, let's understand the two things. Okay, so in the meanwhile, while the page is loading, that your can we upload the raw protein here itself, like the direct PDB we can upload, and the direct ligand molecule from your pubchem and all we can upload? Yes, we can upload, that is not a problem, but mostly we should not directly upload the protein. Why? Because here it is not cleaned for the heteroatom, so we need to clean it from the like for the heteroatoms we need to remove the water, any ligand pre-attached ligands if there are any. Okay, so we need to remove that. Why? Because if there are pre-attached ligands, so they are already occupying the active sites. That is one thing. Second thing is that it might hinder in our process, like whatever ligand we are giving and whatever protein we are coming up with, the interaction that would be happening, that might be hindered by the all other pre-attached ligand. Okay, so that might occupy that particular region. Okay, and other water molecules and all other can influence the docking result. That is why we don't want that, and that is the reason we'll be cleaning our protein from. Okay, so PDB. Okay, so here let's go to PDB, and you can search the protein name as per the disorder you like. Okay, and the protein should be having a what do you say, good role, not good role, basically important role. Okay, so it should be playing an important role in the what do you say, etiology of the disorder. Okay, or likewise. Okay, so let's go on to your 3CL Pro. Okay, so 3CL Pro is the main proteinase of your 3C like proteinase. It is called as, in short, it is called as 3CL Pro. It is a main protease of your SARS-CoV-2. Okay, or COVID-19. So let's look at that. So once we move inside that, so we'll have multiple structures, okay, of that particular protein. So we can go via that, we can download the structure also. Okay, so we can download the structure in PDB, or there are some other ways also. I will also show you some other ways while we are cleaning that particular protein. Okay, so let's suppose this is the protein, that is your 7xjw. Okay, I can go inside this also. I can go inside this and I can hit on download and download the protein structure and then clean it. That is one possibility. Okay, apart from that, I also have one more possibility, that I can go to any of the visualization tool like PyMOL and all. Okay, so once I go to PyMOL, and then I can put this particular PDB ID, load it, clean it, and then save it in PDB format. Okay, so that is what we are gonna actually do. Okay, it is just to show you how to find the protein. Okay, I have demonstrated you this particular part. Okay, so here you can go to download files and basically download the file in your PDB/CIF format. Okay, that is there. So, and one more way of cleaning the heteroatoms is you can go to your PDB format, not by display, obviously you have to download it. Download, then go to this particular PDB format. Okay, and once you go through this, okay, so if you go to the drop down menu, okay, from here, so obviously it is a very long format. Okay, if you go like this, okay, at the end, basically at the end, you'll be finding a HET-ATM. So you can see here it is starting HET-ATM 1 2 3 4. So likewise you need to delete all of the HET-ATM, basically means heteroatom. So this is a short form of that. Okay, so till this particular point you delete all the heteroatoms and save the file in PDB format itself. So this is the PDB format as a whole. Okay, based on this particular formula or the format, this particular protein that you see in the 3D is being curated. Okay, because here you have the XYZ axes. This you can see XYZ axis with angstrom, how much. Likewise at the atomic level, everything is being mentioned over here. So you can download the particular thing I have already stated, or we can use a visualization tool to clean the protein. Okay, so there are two ways. Now let's get on to the pubchem. Okay, I will show you the protein cleaning part, but prior to that, let's take the chemical, because we don't have to do anything with the chemical. Okay, like we don't have to process the chemical. Basically, directly we'll be taking. So that is why we'll directly go on to the pubchem, and I am just giving you an arbitrary example over here. So I will be taking a random chemical molecule. Okay, and we will see that it is having a good affinity score or not. Okay, So, and you can also have background. How do you choose? You choose based on, like, let's suppose you are working on, here we are working on any of the, like, SARS-CoV-2, we can go on to any of the plant phytochemical or synthetic phytochemical which has antiviral property. Based on that, and it is also passing all the pharmacokinetic, pharmacodynamics parameter, drug likeness, then you can proceed with the docking. It is not that easy that you can select any ligand compound and start with the docking. That is not the actual way. Okay, but obviously that is a very long process. I'm just talking about the docking step over here. Okay, so we will talk about that also sooner or later in this particular channel only, so stay tuned. Pubchem, then we go on to this, and we'll be taking a ligand from your Tinospora cordifolia. So okay, so that is a plant. So that is your Tinospora cordifolia is basically a medicinal plant which is having, which has been proven for its antiviral properties. So we'll take one of the phytochemical. Obviously, a particular plant would be having multiple phytochemicals. We need to screen it via all of the things that I have already spoken of. Okay, and after that, whatever phytochemical are like passing all the filters, then we can take it. One of such compound is your tino stenosporide. Here you can see tenosporide. So this one. So we will check our 3cl pro against this one. Okay, 3cl pro is the protein. Okay, 3cl pro is the name of the protein. Okay, but apart the id or the pdb id of that is a bit is different. So that is your 7xjw. So that we will be processing. So don't get confused, this is a protein. Okay, and this is a chemical molecule. Okay, and this is called as a lead. The chemical molecule is your lead or ligand basically, and this is your, the protein is the macromolecule. Okay, yeah. So this is the 3D structure. We need to download the structures in 3D. Okay, so you can go on animate and you can see that how it is moving, although it has nothing to do with the structure download. So you can just hit on download coordinates and hit on your save file. Okay, so why I have minimized this particular tab? Because I need to work on PyMOL also, that is why. Okay, so SDA format it takes into account, so I am taking it, saving it. Okay, so I will hit on save, and here it is saved. Okay, here you can see it is saved already. So it will be saved and downloaded in the path that you have given. So in my so I have given the path of download, so it will be downloaded in the downloads folder. Okay, in my system. So that is there. So now let's get on to the cleaning part. Okay, so for that I'll be using your PyMOL. Okay, so here it is. So you need to install PyMOL. It is very simple. You just need to type in PyMOL in the Google. Okay, and then right away install it, and it will always have this particular what do you say, thing that is written as your just give me 1 second. Yeah, so basically always you will be having this particular thing written over there, that no license for like 0 days are remaining. So don't worry about that. It will run for like many a days. Okay, so like this. Okay, years also. So it is just a disclaimer that they give. Okay, so now we need to upload our like a protein. Okay, so for that we need to do two things. Okay, so it depends upon if you are using a Mac and if you are using a Windows. It is the options where you are getting the options is a bit different. Okay, so here I am getting the options at the top. Okay, because I am using a Mac. Okay, so whereas you will be getting the options if you are using a Windows, and likewise you will be getting the option somewhere. So what to do, you need to go to files. So that is why I'm naming the things. Okay, so you need to go to files. Okay, and then go to get pdb. Okay, so files and get pdb, and then you need to write. So wherever you are having the options of files, okay, you just go to get pdb and type in the that particular id of. Okay, so the id was 7xjw, so that we don't have to download. Otherwise you can download and double click on it and it will be opening it in PyMOL. Okay, that is not a problem. So you just you can write this 7xjw. Okay, so that is 7xjw. Okay, so download. So here I have the protein by my side. Okay, So now I can do multiple things with this. Okay, so I can, for the representation part, I can change its color, this that. Okay, I can like select multiple things. Okay, now you can go to your mouse. Okay, so wherever you are getting the mouse option, and select the chains. Okay, so I want to proceed with only one chain. Okay, so that, for that you need to study the literature. Okay, so which chain is important. Okay, so I will remove all others. Okay, so here we have multiple NSPs like NSP 576. Okay, like and so I will be deleting. I'm just taking an arbitrary example, so I will be deleting all the what do you say, your ligands. So these are the pre-attached ligands that you can see over here. Okay, so pre-attached ligand, multiple pre-attached ligands it is having. Okay, and multiple chains also it is having. So let's suppose I want to proceed with only one chain. Multiple chains are repeated also over here. Okay, so you can also do command or control. Okay, and then delete. That is possible. Hit on remove, and the red dots that you see over here, the red dots. Okay, yes everyone, the red dots are what? The red dots are basically the water molecule. Okay, so that water molecules were being added when they are this particular structures are procured. If it is done using wet lab experiment, you will be having more of water. Okay, like because the experimental requirement is that. Okay, so it is a very large protein, that is why it is taking a bit time. Otherwise it is a snap of a thing. Okay, so just few of the pre-attached ligands and what do you say, chains you need to remove. Likewise. Okay, and water removal is far easier. So you just go to all, you just go to action and remove water. Okay, all of the red dots you can see are vanished because all of the water molecules are cleaned over here. Okay, so these two chains are remaining. Let's suppose I delete this chain also. I just remain. It is not that every time you have to like delete all other chains. You need to remain with only one chain. I'm just giving an example over here. Okay, it depends upon the literature. You need to study that which particular chain is important, and based on that you will be proceeding. Now here you have the clean protein, absolutely clean protein. Here you can see no ligand is attached to it. Apart from that, no water molecule is also there. Okay, then you select the whole thing. You can just like, it is, I'm just showing for your ease. Okay, although you don't have to do this. Coloring has nothing to do with docking. Okay, but just for your understanding, I'm showing that how to color it and like do it in a nice format. And likewise, okay, you can also do go to display and change the colors. Okay, so and the quality of it to max you can do. Okay, max quality. Apart from that, you can go to like a color space and do a publication standard for your publication. And then you can also do a background as white, and likewise. Okay, so we'll talk more about this when we talk about PyMOL demonstration in details in this particular channel. In the future we'll be doing that. Okay, so but most requested video is this particular docking one, so that is why I'm proceeding with docking right now. Okay, now what you can do is, so you just have to save the file. Okay, in PDB format, because that is what is accepted. Okay, so now you go to file. So why I'm telling all the things loudly, because I know that you cannot see this particular files and all over here, because it is in top in Mac. Okay, that is why. And that particular section I haven't selected to screen record, that is why. Okay, so you can just go to wherever you are getting that particular option. We are going to files, then we'll go to your export molecule. Okay, export molecule, whatever is the current state, you keep it as such. Okay, then go to PDB option. You can click on multiple connection, or keep the default as it is. Okay, so once you keep the default as it is, then you can hit on save. Okay, while saving, you can have like you will be having the multiple options. Basically, what is the name via which you need to like you want to save it. Let's suppose I name it as protein. Okay, protein. Okay, protein. And then this is the path, this is my path where I'm saving it. Okay, so I can so this is the proban is the folder name. Okay, and then I have to select. So this is pdb/cif. Don't select this one. The second one you have to select, that is only pdb. Okay, so now I have to hit on save. Okay, so if by the same name I already have another protein by the same name, although that is a different protein, but by the same name I have saved, so I will replace it basically. Okay, so that I can have this protein saved in that particular place. Okay, so now it is saved. Now let's get back to the like this one. Okay, so let's get back to our main exercise. Okay, so yeah, let's stretch it a bit. Yeah, so now what is there? So basically, so this is the DiffDock page. Okay, so that we will go on to. So here it is. So we need to upload our ligand over here, protein over here. Okay, so let's do that. We have processed all the things. Now let's do the end step. Okay, upload new file. We'll go to upload new file, and here is the confor 3D structure is the ligand one. Na? So here I will go on to this and I will hit on upload. Okay, so I will hit on upload. Okay, so the ligand molecule is uploaded. Okay, so let's move this one. Then I will go to your select protein. Okay, so I will go to upload new protein file. Okay, so I will have to search because I have saved it different in the different folder. So I can also go to your proban. Okay, so here it is. Okay, so protein.pdb. So this is what we saved right now, isn't it? So we'll upload this one. So here you can see protein.pdb is uploaded in protein, and this is .sdf format. It is in .sdf format only. It is uploaded our ligand molecule, that is from Rhinospora cordifolia plant. Okay, now generate poses. How many poses I want to generate? Poses means based on the ranking, how many poses it will be giving us. Okay, so I just want top 10. Okay, so top 20, like till 40 you can get. Okay, I want top 10 poses. Okay, so that is why I'm removing it from your to 20 to 10. Okay, then come to diffusion step. What is the diffusion step? Basically, diffusion step, if you increase this one, this is just like exhaustiveness. Okay, the more you increase it, more computational power is required. Okay, so I will keep it to what do you say, 11 is good enough. Okay, although it is backed by GPU, it won't be taking much time also if you like pull it to max also. Okay, diffusion time. Okay, so how much time? So again, keep it like similar. Okay, likewise 11 and 11 we are like both keeping it as 11 and 12 likewise. Okay, so let's see if we can do 11. Yeah, both 11 we have done. Okay, likewise, then no more options is there over here. Okay, we will just hit on run and see nothing is there, because it is now it will be now doing it for our protein and ligand. Okay, so now I will hit on run. Okay, and once I hit on run, so there will be a generate. Here you can see the generate option is coming. So in the background, obviously they are having GPUs and NPUs, like your neural processing unit and graphical processing unit, and supercomputer obviously. So the supercomputers are having GPUs and NPUs obviously. So backed by that, so that is why it can do the things in so fast. Okay, which might take 1 or 2 days. Okay, as a whole, how much rigorous thing what you are doing over here. So but see, in seconds it has given me results. Okay, here you can see all the different these are not different ligands, mind you. That you might be thinking we have just given one ligand, how come it is having so many ligands? It is not ligands, it is all the different poses. You can see I have, like, it is already by default it is clicked on to view all poses. So all of the poses are shown at a particular one stack. Okay, for one particular, this is the active site where multiple, obviously a ligand can fit itself into the same pocket in multiple poses or multiple ways. No, so these are the multiple ways how in what it can fit itself to. Okay, so if I deselect this one, okay, so then I have the top one. See the rank 1, the rank 2, the rank 3. Here you can see the poses are changing. Ligand is the same. This what we have uploaded. Don't get confused. Okay, so here I have already stated the lower negative value is a better score. That is why you can see the minus 3.9, which is a good score in all other docking tools. Okay, here is just the vice versa, or basically the opposite of that. Okay, so here it is just the opposite of that. Here a good score is the what do you say, mathematically what do you say, higher score. That is your mathematically higher also, like it is -1. -1 is obviously bigger than -3 or -3.9. Okay, that is why here we have it. So this is also a very good score. Okay, so here you can see. Okay, so you can also look about the interaction if you want to center it. Okay, apart from that, sideways you can like what we did for your Swiss dock. Okay, after the docking, so that particular thing, here you can see the bonds. Okay, so this particular bonds here also you can see this one. Okay, so these are the bonds, dotted bonds, that how the ligand and the protein are interacting. Obviously, the atoms in the ligand, okay, so the particular atoms in the ligands would be interacting with the atoms of the amino acid in this particular protein. Here you can see, yeah, likewise, okay, and the dotted lines, these are the basically your bonds. Okay, so you can also select on multiple one, or if you want to do it center, so center pose, so it will basically basically zoom in the particular thing, and if you want to reset the zoom, you can go to reset zoom. Okay, and then you can zoom according to your convenience, whatever you feel like. Okay, and then likewise you can see, okay, you can maneuver this in whatever ways you want. Okay, yeah, so yeah, so that is it. Okay, so that is about it, and that is about today's demonstration as well. Okay, so if you find some value in this particular session, okay, and you are able to learn something new, okay, so please don't forget to like the video, okay, share it with your peer ones, okay, and whoever is in need of this particular thing, or whoever is wanting to get started with the field of bioinformatics or AI and want to learn basics of docking, and likewise, or if you are a biotech person also, or any part of life science, you want to get started with your bioinformatics or AI, okay, you can get started with this particular stuffs. Okay, that is there. Okay, and if you have any question, comment down below. Okay, and don't forget to hit the like button, share, and also subscribe. Okay, so we are having very less subscribers as of now, because it is just 4 or 5 days that we have started the channel. So I want your support for this particular channel, so that we can continue making more of such videos. Okay, and we can provide it for free. Okay, so and knowledge should be for free. Okay, but obviously to a certain extent. Okay, and we are trying our level best to get onto that. Okay, so with that particular note, I'm Pradul Banerjee signing off. Okay, see you, take care, bye bye.